my pleasure to introduce today's speaker for our science showcase. So Dr. Aldaz is a professor in the Department of Epigenetics and Molecular Carcinogenetics at the University of Texas MD Anderson Cancer Center. Dr. Aldaz received his MD and PhD from the University of Buenos Aires Medical School. Dr. Aldaz was among the earliest to utilize high-throughput sequencing approaches for functional genomic studies. The Aldaz lab was the first to discover and clone the WWOX tumor suppressor gene, the target gene of chromosomal fragile excite FRA16D. Dr. Aldaz is currently exploring how WWOX LoF function mutations are mechanistically related to cerebellar ataxias, including SCAR12, neurodevelopmental pathologies, and neurodegenerative disorders. So with that, I'll turn the floor over to Dr. Aldaz.
Thank you, Celeste. Let me start sharing my screen. Just a second, please. I was doing the wrong thing. All right. Can you see my screen?
Yes, we can.
All right, good. Thank you so much, Celeste. Thank you so much to the NAF, the National Ataxia Foundation, for giving me this chance to present in this showcase. I will be talking about, the title is a little bit different to the one that you were viewing, so it's more targeted to the talk in particular. So I will be speaking about this gene that was discovered in my lab several years ago, many years ago, actually, which we named WWOX, WW domain containing oxidoreductase. We name it that way because it's a gene that has two WW domains. The protein has two WW domains, which are protein-protein interaction domains that have a predilection to bind proline-rich motifs, in particular PPXY motifs. And also it's an oxidoreductase, it's an alcohol, the alcohol dehydrogenase family. It has a catalytic site, a well-known catalytic site, and a cofactor binding site for NAD or NADPH. The protein itself tends to localize in a perinuclear compartment, in particular overlapping quite a bit with Golgi or endosomal endosomal reticulum compartment. The gene in particular is a huge gene of over 1 megabases in size, and at chromosome 16q. And we know now that we knew then when we cloned it actually, that it contains or spans what it is known as the chromosomal fragile site 16D. So it has one of the largest introns known in the whole genome. This intron 8, it has approximately 800,000 base pairs in length. What are fragile sites? Fragile sites are specific loci that are prone to gaps and breaks in the genome, and in particular we all have them, and they are exposed. This breakage or rearrangements, it usually takes place when the cells are exposed to mild replicative stress. These are some of the insults that can lead to breakage in these sites. This fragility is conserved through evolution, and we can see in mouse is exactly the same as in humans. WWOX contains also a fragile site, in this case in chromosome 8E of the mouse. And it's also a large gene in the mouse as well. There chromosomal fragile sites genes are extremely large. In general, they replicating, and fragile site 16D is the second most common site for breakage and rearrangement in the whole genome. And this breakage and rearrangement is important not only in somatic alterations, like in the case of cancer, but also, as we will see, in germline alterations. That's how we originally discovered the gene. We were focusing in breast cancer and looking for areas where loss of heterozygosity was common, and we ended up cloning the gene. As you can see, here is a list of various tumor types, and I detour in a little bit to the cancer arena, in which we know that the gene undergoes translocations, deletions, and loss of expressions by multiple mechanisms in these various tumor types are the most commonly affected. And usually the most common abnormalities are deletions, deep deletions. Here we have several tumor types that are highly affected by deletions of this target gene. Somatic mutations, point mutations, are not that common, since this is, for instance, information gathered from over 10,000 tumors that can be found in Tumor Genome Consortium. Now, this is an example of translocations. One of the diseases that have translocations affecting this gene, somatic deletions, somatic translocations, translocation 14, 16, and these are some of the multiple breakpoints, in particular in exon 8, in which here the immunoglobulin enhancers come over and translocate to this region, and it is supposedly to activate this oncogene that is 3' from WOX, a small oncogene called NAF, and in theory, the immunoglobulin enhancers activate the expression or overexpress this particular gene, but in the process, we are losing a copy, or we are losing copies of WOX as well. Now, it was far more recently that we know that germline, in this case, germline pathogenic variants were discovered either as compound heterozygous, compound mutations, or in various neurological disorders. The first one that we discovered, that was discovered, in which we participated, is a spinocerebellar ataxia type 12. And we will be talking a little bit about WRE, which is WOX-related epileptic encephalopathy. And now we know that WOX is also a risk gene for various common neurodegenerative conditions like Parkinson's disease, multiple sclerosis, and Alzheimer's disease as well. WOX is expressed all throughout the central nervous system, in particular, more highly expressed in cerebellum. As you can see, this is data from the gene GTEx, but it's all in all regions of the brain. We can find it expressed, and this is information throughout the life of humans. And you can see that the red line portrays a cerebellum expression, cerebellum cortex, cerebellar cortex expression, and it is higher in early stages of life, in which cerebellum takes the largest development. It is expressed also in all cell types in the central nervous systems, neurons, microglia, astrocytes, and oligodendrocytes as well. And we know now that also it is a gene that can be induced, at least in microglia, with, here's an example of injecting lipopolysaccharide to mice, and the expression of WOX, it is increased. In terms of particular brain regions, of more specific brain regions, and this is information from the Allen Mouse Brain Atlas, we know that basolateral amygdala, medial entorhinal cortex, this is basolateral amygdala over here. These are in situ hybridization studies done in the Allen Mouse Brain Atlas. The entorhinal cortex, the cerebellum cortex, and also not shown here, the isocortex layer 5 in particular. And these are the main functions of these regions, like basolateral amygdala, memory consolidation, fear response, and entorhinal cortex is a very important space where that connects basically the hippocampus with many other regions of the brain. This is a very recent information in which which is not published, and in collaboration with Xiaolong Yang from Baylor College of Medicine, in which single-cell RNA sequencing, these are various clusters expressing various markers of different brain regions, and we can see that WOX is mostly expressed in this area, and this area, this cluster, overlaps quite significantly with the cluster that expresses the marker parvalbumin. I'm sorry, the cluster parvalbumin. Parvalbumin is obviously a marker of interneurons, inhibitory neurons, GABAergic neurons. Here we are looking at two markers of cerebral cortex, of isocortex, of the various layers from 1 to 6. And this marker that is expressed throughout, and there's a marker here, KCN2, that is expressed mostly in the deep layers of the cerebral cortex. And there is quite an overlap from WOX location in these deep layers of cerebral cortex. So our assumption right now, at least in isocortex, is that WOX is expressed in deep layers of isocortex, overlapping with parvalbumin enriched cells, interneurons. So this was the first study in which we discovered, we participated in this study, in which it was discovered that WOX was associated with cerebellar ataxia and was associated in general with central nervous system conditions. These were a family, a Saudi Arabian family, in which both parents contributed a mutation, it's a germline mutation, the proline 47 threonine mutation, and this is obviously a consanguineous family, and these 4 siblings developed this condition, which was named autosomal recessive spinocerebellar ataxia type 12, in which these kids have seizures, epilepsy, delayed psychomotor development, spasticity, significant intellectual disability, ataxia. And this mutation is located, there was another family that was also discovered, the G3372 mutation, but this particular mutation, it is sitting on the WW domains, which are important, as I mentioned earlier, for protein-protein interactions. But we can consider these mutations hypomorphic mutations, in which there is partial loss of function. These individuals express still WOX protein, but the protein is mutated, and very likely still the enzymatic domain is working, while in this case, or in this particular family, we know that the protein is unable to bind other proteins that have the proline-rich motifs I mentioned in an earlier slide. Now, soon after that, we saw these much more severe conditions in which these are kids with that display refractory intractable epilepsy, severe developmental delay, not able to speak or sit or walk, movement disorders, spasticity, in the more severe cases, spastic quadriplegia, even in some of them, severe structural defects in brain, some even have microcephaly, eye development abnormalities, and in the more severe cases as well, early death. These cases were described as WOX-related, this is the other name given to this condition, is WOX-related epileptic encephalopathy, or WRE. In this case, contrary to the previous one in which I mentioned that were hypomorphic mutations, here these are the mutations provided by the parents. Again, this is recessive. Provided by the parents could be compound heterozygous mutations mostly, lead to complete total loss of function. These are null mutations, either a combination of stop codon mutation with a deletion, or a combination of 2 missense mutations that lead to total loss of function, or a nonsense mutation. This, one of the first experiments we originally did was the development of WOX knockout mice. The WOX knockout mice basically emulate or mimic quite closely the WRE condition that I just described in the previous slide. You can see here in this case, I'm inducing seizures in these mice by exposure to sound, to a high frequency sound, but the mice also develop spontaneous epilepsy seizures constantly just by handling. They have also growth retardation and a short lifespan, ataxia as well, obviously, and a short lifespan of only 3 weeks or 3 to 4 weeks maximum. And you get the picture. They get all mice get into seizures. The brain of these mice have in the first studies we were focusing in hippocampus, and we noticed in hippocampus a loss of interneurons, in this case parvalbumin positive interneurons or MPY positive interneurons. That was one of the observations, and we also noticed a decrease in this is a Western blot looking at the enzymes that are critical for the synthesis of GABA, and we see a decrease. This is showing that these are full knockouts, and we see a decrease of expression of these enzymes in charge of synthesis of GABA. GAT64, GAT1, GAT2, or GAT65, 67. We also noticed that there was neuroinflammations in the brains of these mice, here shown by positive staining with IBA1 and the characteristic phenotype of the cells that are activated by microglia that is activated. And here we see evidence of astrogliosis as well by GFAP staining. We see more astrocytes and activated astrocytes in the brains of these mice. So we know this is one well-known review regarding autosomal recessive cerebellar ataxias, and the genes involved in the various ataxias touch several common themes like mitochondrial metabolism, DNA repair, lipid metabolism, and indeed WWOX. There are reports of WWOX of basically playing roles in all these conditions. We did quite a bit of work in DNA repair, but it's also, there are papers that demonstrate the role of WWOX in mitochondrial metabolism and lipid metabolism. So it is, these are the 3 main clusters that were identified as targets of multiple cerebellar autosomal recessive ataxias, or ARCAs, like are named here. As I mentioned earlier, also WWOX has been identified as a risk gene for various common neurodegenerative conditions, and one neurodegenerative condition that is clearly linked with ataxia is Parkinson's disease. And WWOX is one of the genes that was discovered recently in this meta-analysis as a progression loci of Parkinson's disease, a progression locus of Parkinson's disease. Multiple sclerosis, another condition that is linked to ataxia as well. WWOX has been also identified. There are multiple that map to WWOX, and that were shown to be significantly associated with multiple sclerosis. And ultimately, Alzheimer's disease also is one of the new 5 loci, genomic loci, that have been described in the largest meta-analysis to date of Alzheimer's disease. Okay, so going back to that first family I described earlier, we now generated a knocking mouse model by CRISPR-Cas9 technology, in which we introduced the exact same mutation that we saw in those 4 siblings that I was mentioning earlier, in affecting WW domain 1 and affecting WW domain now. So we are mutating a proline to a threonine, and that's the only amino acid we are changing the whole protein. That mutation is sufficient to inhibit the ability of WWOX of interacting with other proteins. When we have that mutation, WWOX that cannot interact with proteins that have these proline-rich motifs. This is an example of 2 common interactors of WWOX, like DBL2 and WEP1. And as you can see, this is from cerebellum extracts, and you can see the interaction is abolished by the mutation. And these are cerebellum from homozygous mutant mouse versus wild type mouse. So we now have this knock-in mouse, contrary to the full knockout mice that I showed you earlier, that within 3 weeks, these mice live a long life, even though they have a decreased, reduced survival compared with the wild types and the heterozygous animals. These animals display spontaneous seizures. Here is an example of one mouse that is seizing, spontaneous seizures, but are not lethal. So they live with seizures constantly, and some are not severe, but they have them every day, multiple times. And that's likely why they die a little bit earlier. We subjected these mice to various behavioral and motor studies. In this case, this is the open field test, when you place the mouse in an arena. And as you can see, this helps to, consider a test for measuring anxiety, willing to explorate, and general locomotor activity. As you can see, we are tracking the mouse with, in this case, with the ethovision software, and you can see that the mouse moves all over, the wild type mouse, the heterozygous mouse as well. But in the case of the P47 homozygous mutant, we see that the mouse does not explore as much and remains mostly in areas that are close to the walls of the container. And here simply is describing in numbers what we have observed here. The mouse traveled much less distance than the wild types of the heterozygous animals, and remains less in the center area of the arena. In the Rotarod test, another critical test for evaluating motor function, we see that mice that are homozygous for the mutation display evidence of the latency to fall is much shorter, and they travel less distance as well. And also we did other tests of ataxia, like the tracking of the walking of the mice, and they get to a point in their life, in the life of these mice, that they basically become immobile. They just stay on one corner of the cage and don't move for a long time. They just move to eat, and that's it. Another test is the elevated plasma maze. This is a typical test also to measure aversion, anxiety mostly, and we see that the mice tend to remain in the enclosed areas of this test and not to go on the open arms, and they are less in the center area of the maze. We did also tests of social behavior. This is the typical juvenile social exploration test. And again, the mutant mouse spend less time visiting or interacting with other mice, as we can see here. But the gold standard for measuring social novelty and social interaction is the 3-chamber sociability and novelty test. As you can see here, mice are placed in these chambers, first in the center chamber and these 2 little chambers on the sides. First, we put one mouse that is called the novel mouse, and then the familiar mouse first, and the normal mouse goes and explores the mouse around. These are like little jails where the mice are sitting. And then when we place a second mouse, the test mouse goes and explores both of them, but most often goes to the novel mouse than the familiar one. And this is tracking of these animals. And in here, these are the little jails that I was describing. And a wild type mouse, you see, it goes to the familiar mouse and then explores the novel mouse. And this is the heterozygous mouse. It does the same, tracking the movements of the mouse. But as you can see, the mutant mouse stays completely out of interacting with the either novel or familiar mouse, and just stays exploring just the periphery, not exploring, moving around the periphery of the cage, and does not interact. This is sort of a standard for evaluating autism type behavior. When we look at the brains of these mice, in this case in the hippocampal region, we observe that there is significant neuroinflammation. In this case, we are evaluating the mice at 80 days of age and 250 days of age. And we see that this neuroinflammation increases a bit with time. In particular, and these are measurements of various parameters demonstrating that these are indeed activated microglia. So the neuroinflammation, it appears to be progressive with age. The same is for astrogliosis. These are GFAP stainings in which we are looking at astrocytes, and we see activated astrocytes that increase with age. And it is quite dramatic the way that the astrogliosis develops in these mice with time. Here we are looking at oligodendrocyte numbers and staining of another staining for myelin production. And we see a decrease in numbers of oligodendrocytes in this case in corpus callosum of these mice. As you know, myelin is critical for conductivity. We did transcriptome analysis in the forebrain of these mice first, and we saw basically confirming the observations that we were, the microscopic observations that we were seeing, alterations in glial differentiation, in hippocampus development. These are activated pathways or ontology terms, and these are suppressed ontology terms. Among the suppressed, we see glial differentiation suppressed, hippocampus development suppressed, and cerebral cortex development suppressed, forebrain development as well. And in this case, we are looking at, this is from hippocampus tissue, and this is from cortex, cerebral cortex. And we see interesting, although we are analyzing cerebral cortex, we see evidence of hindbrain development suppressed, as can be seen by these clusters. Cerebellar Purkinje differentiation suppressed as well. So this evidence, even by analyzing the transcriptome of hindbrain, of forebrain, that in hindbrain something was going on. And the pathway analysis basically confirmed what we were seeing microscopically of activation of multiple inflammatory related pathways, in particular, and also neurogenesis pathway. Like one example is CREB1 activation in all these regions of the brain, hippocampus, cortex, and prefrontal cortex, in which we see perhaps in an attempt to compensatory neurogenesis. And these are pathways that are also activated, as I said, TNF-alpha, interleukin-C inflammatory response, hypoxia, apoptosis. Now, seeing the abnormalities described when the motor abnormalities are described, we look at hindbrain, cerebellum. Even microscopically, we can see this is a wild-type sample, and these are knockout mutant homozygous samples, and you can see that the cerebellum, even macroscopically, looks hypertrophic, and we don't see the vermis is not as developed as in the wild type in these mice. And indeed, when we measure cortex, cerebral cortex, we see decrease in the thickness of the molecular layer and the granular layer of the cerebellum cortex. When we go into more detail, we indeed see a dramatic decrease in the number, the staining, in the red staining with cal binding stains, in particular the body, the soma and dendrites, and of Purkinje cells, which can be seen here, a low magnification, a high magnification here. and you can see how they are lost. We lost the staining throughout, and we clearly lost staining. We see the somas are not there, and we see dramatic abnormalities in the dendrite of these cells. Here we have a second staining with green. We are looking at basket cells that are sitting on top of the Purkinje cells. In the wild types, we can see one Purkinje cell, one basket cell, one Purkinje cell, one basket cell. Interestingly, here we can see the basket cells are there, but the Purkinje cells are not there. So the number of Purkinje cells decreases and it decreases further with time, but the number of basket cells remains the same. When we look at the gene expression, confirms that some of the pathways are related to hypoplasia of the organ, congenital neurological disorder, familial congenital malformation, encephalopathy, motor dysfunction. And these are some of the pathways also that are overrepresented at the transcriptome level. These include the apoptosis, inflammatory response as well. And we confirm the evidence of neuroinflammation as well. Now, one of the additional studies we did is called RPPA. RPPA is a technique that, for those that are not familiar, basically is to do Western blots with from cell extracts. In this case, we were using cerebellum from wild type animals and cerebellum from mutant animals. We did protein extracts. They are array, and then these arrays are blotted in our case here. I don't know what happened with the references up here in the middle of the slide. The reference of the originators of RPPA, the developers of RPPA, in which these arrays of tissue arrays are blotted with. In our case, we did approximately 400 different antibodies of various pathways, and conditions. Here we see this is the expression, the relative expression, which is all stringently controlled for intensity, for loading, for everything of the wild type versus the mutant cerebellum extracts. We see activation of multiple genes that have to do, again, with inflammatory response. In this case, we are focusing on cerebellum exclusively. We also see activation of the notch pathway, activation of notch clip. This could be perhaps related to astrocyte differentiation and also confirming the evidence of astrogliosis, because notch gets activated also in the adult in astrocyte differentiation. We see evidence of apoptosis. Various apoptotic related genes are overexpressed. Not genes, in this case, proteins are overexpressed in the cerebellum of the mutant mice. And we see also evidence of necroptosis. So it is cells are dying, basically. That's the bottom line story in the cerebellum, as well as in other regions of the brain. This has to do with the cell cycle arrest. And ultimately, and I'm showing some, as I said, that we have information for 400 different proteins on the brain. For different proteins that we look at and various pathways. And CREB activation, in this case phosphorylation 133, which probably again could be compensatory neurogenesis. So maybe I went too quickly, but the conclusions we have here is that our knocking mouse model, the one we just developed, the WOX P47, is phenocopies almost identically human cerebellar ataxia type 12, because we see seizures, we see ataxia, and we started investigating what is happening in the brains of these animals. We know, and besides, we have abnormal behavior, impaired motor coordination, as I described. We see progressive neuroinflammation, cerebellar hypertrophy, Purkinje cell degeneration. And this mouse model will be useful not only to study what is happening in this rare condition, if you will, but also it will be useful to look at more common neurodegenerative conditions that in which WOX have been identified as a risk gene, like I described, like Parkinson's disease, perhaps multiple sclerosis, and indeed also Alzheimer's disease. At least in Parkinson's disease and multiple sclerosis, we know that ataxia is a critical component, a clinical manifestation. In future directions, we will be soon starting to test the potential of gene rescue by utilizing, I don't know, AAV delivery. This has been successful, at least in full knockout mice, in stopping the development of epileptic seizures. We want to test whether delivery of WOX by this means, either by ICV injections or also we'll try intrathecal injections, although we don't think that will be successful. But in ICV stereotactical injections in the brain of these mice will be able perhaps to inhibit the cerebellar neurodegeneration that we have been seeing, and the degeneration of Purkinje cells as well. We are also developing, planning to develop Purkinje specific knockout mouse models to see whether we can determine whether indeed Purkinje cells are the target cell and the reason why these animals develop the cerebellar hypertrophy and the ataxic conditions. So I want to acknowledge Tavish Hussain from my lab is the one who have done most of the work, collaborator from University of Texas at Austin, Baylor College of Medicine, University of La Plata and Argentina, and also the various cohorts that participate in this, obviously the funding from the National Ataxia Foundation and other sources of funding for these studies. So I will stop there and will answer any questions. And maybe, as I said, maybe I went too quickly, but no, we have time for questions.
Yes, and thank you for such a fascinating talk, Dr. Aldaz. Just a reminder for our attendees, if you have any questions, you can put them in the Q&A window at the bottom of your screen, or if you'd rather ask them directly, you can use the raise hand feature on Zoom and we can call on you. And while we're giving attendees a few minutes to formulate their questions so they can bring in, we can start with a question of my own. So at the beginning of the talk, you had some data about the expression of WOX throughout a person's lifetime. And sorry if I missed this, but is there any information available about why WOX is so highly expressed in childhood and adolescence as compared to other stages of development?
Well, obviously in childhood, at the beginning in the early life, the brain is developing and cerebellum is developing too. So in part, that is the reason why we see an increase early in life for expression of WOX. But cerebellum always runs above the other ones. Obviously, cerebellum is a critical organ for controlling, or it's the control center for movement, for movement control, for voluntary movement, speech, vision, posture. So it's critical in the development of the baby, when the baby starts, sitting and moving, and so we know the neurons keep being produced at that time. So I don't have a good answer for your question.
Oh, no, that's very helpful clarification. Our next question was regarding some of the anxiety phenotypes that you saw in your mouse model. So it's a two-part question. First, are those phenotypes seen in human patients with spinocerebellar autosomal recessive 12? And is there any work being done by neuropsychologists about how to potentially help treat these symptoms?
Well, in the case of SCAR12, the spinocerebellar ataxia, those patients have severe intellectual disability. They have originally were described mental retardation. That's not a politically correct term now, intellectual disability. And I don't know if they have that particular set of siblings, if they have autism type of behavior. But yes, indeed, and something I didn't mention, WOX deletions have been seen in autism families as well. And if autism is one of the conditions in which WOX is affected by germline deletions in particular, and mutations have been seen in simplex families, in families with one kid, but also in multiplex families in which deletions have been observed. This is important, and indeed, in the case of the total loss of function, the worry syndrome that I described later, yes, some of those kids, because it is not black or white, it's not this disease or that disease. It is a spectrum of conditions. And the more severe the condition, obviously we see those kids. The dramatic pictures I show there, those are the most severe cases, but there are kids that are not as affected. And yes, they have signs of autism, autism spectrum disorder. They have social interaction issues that have been demonstrated, no doubt about it, yes.
Well, thanks for clarifying that. Our next question comes from an attendee who is interested in knowing if any of your findings or research on WOX has any connection to the vagus nerve or anything in that area.
To what?
The vagus nerve.
Vagus nerve?
Yeah.
Not that I know, no, honestly, I haven't looked at that, and I don't know if anybody has, no.
That's A-okay. And then moving on to another one of my questions. So your mouse model phenotype, it's homozygous for the P47T mutation. Is that the same mutation you found within the family, or was it mainly heterozygous within the human patients and then homozygous in the mouse model?
I'm sorry, can you repeat the question? Because I was reading something and I got lost.
That's A-okay. For your mouse model, it was a homozygous mutation. Sorry if I missed this, but was the homozygous mutation also present within the family, or
yes,
was it
absolutely, yes. No, it was a homozygous mutation. These were two cousins that got married, and one had the P47T and the other one had the same mutation, P47T, and the kids, that's why some of the kids inherit the mutation, homozygous. There was a family like with 7 children, with 7 kids, of which 4 have inherited the mutation, from both parents, and they have the condition, spinocerebellar ataxia type 12, and the other 3 did not, because they received the wild type allele from both parents. But even if they receive 1 wild type and 1 mutant, they were going to be normal because the parents are normal, or apparently normal. Of course, I don't know how much clinical history is available in the heterozygous mutant individuals. Yes, there are multiple individuals that have mutations. They have, for instance, deletions of WOX in 1 allele, and they are considering controls or normals, because now by looking at databases, we can see there is a lot of people that have deletions or duplications in WOX, 1 allele, and they are considered controls. But that's why perhaps this is telling us why WOX probably is, we're seeing now that is involving other conditions like common neurodegenerative conditions. It will be important to study this population throughout time, either from the spectrum, from what I said about autism, to also to conditions like Alzheimer's disease, Parkinson's disease, and multiple sclerosis. There is a question here, and question and answer. This is the first time I heard a SCAR type that is autosomal recessive. Yes, no, the ataxias can be autosomal dominant or autosomal recessive. Autosomal dominant is obviously, as you know, is inheriting 1 copy from 1 parent. We will see in the progeny, in the kids, we will see the same condition an example is Huntington's
Mhm.
disease, multiple other SCA, spinocerebellar ataxias. That's why these are not called SCA, these are called SCAR. SCAR
Yeah.
comes from recessive. Yes, in this case, we don't see it in the parents, we see it in the progeny. But yes, there are many spinocerebellar, there are many recessive ataxias.
The attendees on the call may have also heard them referred to as ARCAs, so autosomal recessive cerebellar
ARCA.
ataxias.
Right, correct,
Yeah.
ARCA, yes. It was in one review that I put.
Yeah, it's different terminology meaning similar conditions.
ARCA,
It can be hard
correct.
to keep track. And then the next question here, someone is an attendees thanking you for your presentation. It's a wealth of information. You mentioned that WWOX plays a role in other movement disorders, specifically MS and Parkinson's disease. Does your research show any relation to hypertrophic olivary degeneration, acquired ataxia as a comorbidity of HOD?
We don't know. I don't have a good answer for that. Right now, is with the findings, and I confess I'm not a neuroscientist. I was not a neuroscientist all my career, and now we are getting to study in more detail the cerebellum in particular and the connections of the cerebellum. We are planning to study climbing fibers and how they connect to the deep nuclei. That's in the folder of things to do, because we need to understand what is going on in cerebellum and their connections. But yeah, that's to be studied.
Yeah. Future directions. Speaking of future directions, you did an excellent job explaining the pathogenic effects of WWOX loss of functions on the cerebellum, and you briefly mentioned some of your next steps are going to be trying to rescue those phenotypes. Can you tell us a little bit more about the AAV you're going to be studying? Well,
the good thing about this attempt is that this adenoviral vector has been first tested and developed by a colleague in Israel, Jerusalem Institute, and they basically, as I mentioned, they rescued the phenotype of mice that have the WRE condition, and those they injected in their brains the virus, and they basically cure the mice. So they claim. And they cure the mice. So the mice were able to grow, develop, and even have, although I said that they tend to die within 3 to 4 weeks, 2 to 4 weeks, but they live a longer life, and they are even able to reproduce. So we will be using the same virus that is driven, the expression of the virus is driven by synapsin promoter. The synapsin promoter is going to be expressed in neurons in particular, and eventually in the future we can try other promoters for its delivery. And this virus is being currently produced in large quantities as well, because they are planning to start clinical trials, at least for the WRE condition in particular. So we will test the same virus that is going to be delivered to us. I have a question here. This how will the AAV be delivered? Will it be directly injected into the cerebellum? Well, in the first attempts, we will start with stereotactical intracerebral into the ventricles first. I know that's maybe not ideal, but we will also do it by intrathecal injections. Yes, we need to think also on what are the best approaches to reach cerebellum in particular, but we will start with those two approaches. I'm open to suggestions on how to better address inoculations to reach more proficiently cerebellum if somebody has knowledge or ideas of how to do it. But indeed, those two routes should be able to reach cerebellum by by the CFS. And obviously, some are more efficient than others, and the problem is how much virus reaches the destination you wish to go.
It's one of those ongoing questions in the field of trying to deliver things efficiently and safely. And then one final question from me. You mentioned those two initial families with SCAR12 who were described in your brain paper. How did you initially connect with those families in order to do this research?
Well, this is was a collaboration with a laboratory in France and Germany, and they were studying. They had identified this condition previously. They didn't know. They had pinpointed that the gene was in chromosome 16, and they have this family identified. And then they contacted us because they knew that we were studying WOX, and we have mice. And then basically we confirmed that indeed in the mice we have the same or very similar phenotype to the one that we were seeing, that they were seeing in humans. Although in our case, the mice, as I mentioned, reflect more worry, the more lethal condition, than the SCAR, the spinocerebellar ataxia recessive, in which the mice survive a longer life. The humans survive a longer life, I'm sorry. But yes, they were the ones who have pinpointed the family, identified the family by colleagues that were also in Saudi Arabia. And it didn't have a diagnosis. They didn't know. They pinpointed that was in chromosome 16 in a paper originally, and then with exome sequencing they were able to pinpoint that the target was WOX, and then we identified the P4070 mutation, and then we basically reproduce the findings now in the mouse model.
And the rest is history. Well, I'm not seeing any other questions in the chat. So thank you again so much, Dr. Aldaz, for a fantastic presentation and answering our questions. We really appreciate you sharing your findings with us. And thanks to all of you who attended the session today. I hope you learned something important and that you have a wonderful rest of your day.
Thank you so much, Celeste. Thank you so much to the NAF for support for our studies and all of those that are working in the area. Thank you.